Hello, everybody. Thank you for taking the time to join us for this webinar where we take you through how GenScript was able to rapidly identify a diverse range of high-affinity SARS-CoV-2 monoclonal antibodies in just one day. My name is Grace, and I am the Product Manager for Reagent Antibody Services at GenScript, and I will bring you through the first part of the webinar. In the second half, my colleague Win, our Senior Field Application Scientist, will take over to tell you a little more about how antibodies can be truly optimized for your research application. During the webinar, please feel free to type your questions in the chat box for us to see, as we will be taking some time to answer your questions at the end of the webinar. However, if we do run out of time or are unable to get to your question, we will certainly follow up with you via email. So without further ado, let's begin. During this webinar, I will provide some background into the rationale for this rapid antibody discovery campaign, and I will walk you through our process of how we were able to rapidly isolate, screen, and characterize a large range of SARS-CoV-2 antibodies within 24 hours. Then I will walk you through the results of our antibody discovery campaign by showing you some of the characterization we obtained for our top antibodies. Then, as I mentioned before, my colleague Win will jump in to tell you more about antibodies, such as the ones I will be showing you today, can be further optimized for your specific research application. He will then wrap up this session with a brief summary of what we have covered today. So let's begin. It is without a doubt that the pandemic has impacted and continues to impact billions of lives around the world, with over 28 million people infected, close to 1 million deaths, and a projected cost of 27 trillion to the global economy. It is difficult to fathom a return to the normal workings of everyday life until effective solutions are found to combat the virus. And you might be wondering, what do some of these solutions look like? Well, we need effective diagnostic solutions to help determine who is infected with the virus, to help facilitate treatment decisions, contact tracing efforts, and herd immunity evaluation. We also need therapeutic solutions to help treat individuals sickened by these diseases, to help cure them and better manage symptoms of this disease. We also need preventative solutions like vaccine development, a hot topic today. That is without a doubt one of the most important undertakings to build population immunity against this virus. And finally, we need to conduct research to expand our basic knowledge of SARS-CoV-2, knowledge like its structure, function, and evolution, because this will go really far, not just in informing future diagnostic, therapeutic, and vaccine development efforts, but also in helping us understand how viruses like SARS-CoV-2 arise in the first place, so that we can work actively to prevent such a pandemic from taking hold of our world in the future. So developing all these solutions are important, but often requires the use of specific high-affinity research antibodies towards SARS-CoV-2. And, these antibodies form an important component in a wide range of research assays. Research assays such as virus neutralization tests to detect the presence of neutralizing antibodies in a sample, immunohistochemistry, ELISA, Western blots, binding and cell-based assays, flow cytometry, and many more immunoassays all require the use of monoclonal antibodies, be it as research controls or as lead candidates for diagnostic and therapeutic development, such as the ones shown here. As a result, as the global research community has stepped up to develop COVID-19 solutions, GenScript saw that we needed also to quickly respond to the need for high-quality research antibodies towards the coronavirus. While GenScript has generated a wide range of monoclonal antibodies towards the nucleocapsid protein and spike protein extracellular domain of SARS-CoV-2, in this webinar specifically, I would like to focus on how we were able to rapidly develop antibodies towards a critical part of the virus, the spike protein's receptor binding domain, also known as RBD, as shown here in the left panel. The SARS-CoV-2 spike protein is a membrane-anchored protein that decorates the surface of a virus. And the binding of the coronavirus via its RBD to the ACE2 receptor on a host cell is the critical step that enables viral fusion and subsequent infection of the host cell with the virus. And as a result, given the important function of the spike RBD protein in viral infection, the RBD has become a target of great interest for diagnostic, therapeutic, and vaccine development. And thus, we saw the importance of generating high-quality antibodies to facilitate research in these areas. But given that there are a number of antibody generation methods available, which is really the best to choose from? We know that during this incredibly pressing time, generating a variety of highly specific, high-affinity antibodies quickly was of utmost importance. However, being an experienced custom antibody vendor, we are also extremely cognizant that not all antibody discovery technologies are created equal. Hybridoma technology, for example, is a great, reliable antibody generation method developed in the 1970s. And despite being a traditional method, it's incredibly popular and is still widely adopted today. And while it's a very solid approach for antibody generation, hybridoma antibody screening takes a while, around 3 months. And despite that, seeing that these antibodies are generated in an immunized host, however, despite the length of time it takes to screen, these antibodies are great because they possess natural heavy and light chain pairing, and antibody affinity is usually high. However, with hybridoma technology, antibody diversity, or the number of unique epitopes that these antibodies recognize, tends to be fewer due to the significant loss of antibody repertoire that takes place during the immortalization process known as electrofusion. Another well-known method for antibody generation are display libraries such as phage, yeast, or ribosome display, which are an entirely in vitro way of generating monoclonal antibodies, where antibody sequences originate from DNA libraries originally derived from naive or immunized sources. Screening time for display libraries tends to be a little faster than hybridoma, around 2 months. However, the downside to display libraries is that natural heavy and light chain pairing is often lost, and binding affinity loss is also possible during the isotype switching process. And as a result, affinity for antibodies discovered through display methods tend to range from low to high. Additionally, the screening and selection process from using display libraries is also susceptible to affinity bias. The latest in antibody discovery is actually single B-cell screening, which is a superior technology that essentially overcomes the limitations of these other two methods. Single B-cell screening starts with B-cells from an immunized host, and screening is much more rapid compared to hybridoma and display libraries and can be completed in as fast as 1 day. As these antibodies are naturally generated in an immunized host, natural chain pairing is maintained, and high-affinity antibodies are obtained using this method. Additionally, another key advantage of this method is that antibody diversity is preserved, meaning that it is possible to achieve a range of monoclonal antibodies that recognize different epitopes on the target, giving researchers many options for identifying the ideal monoclonal antibody assay, a monoclonal antibody that works best in their assay. And so compared to other methods, single B-cell screening provides the highest probability of achieving your ideal monoclonal antibody clones in the shortest length of time. And so at GenScript, how do we conduct single B-cell screening? Well, we actually possess the latest in single B-cell screening technology through the Beacon platform from Berkeley Lights. This technology harnesses the use of optofluidic technology that enables high throughput automated screening and selection of B cells on a chip. This means that the Beacon platform is able to accelerate B cell selection and capture a bigger B cell repertoire than hybridoma technology and display libraries are able to. So, as mentioned before, antibody screening in a typical hybridoma workflow takes around 3 months. However, using the Beacon platform, all the cellular import, screening, and export can be conducted within 24 hours. Furthermore, as I mentioned before, through the immortalization process, over 99% of the original B-cell repertoire when using a hybridoma approach is lost due to the unavoidable inefficiencies of the electrofusion or immortalization process. On the other hand, with the Beacon platform, over 99% of the original repertoire is preserved, as these B cells are not subject to electrofusion, but rather are enriched for and then imported directly into the platform. And so what does GenScript's single B cell antibody discovery workflow look like? Animals are first immunized in phase 1, and after anywhere from 2 to 10 weeks, their tissues such as the spleen, lymph nodes, and blood are removed, and antibodies secreting cells are isolated and enriched. The antibodies are then loaded onto the Beacon platform chip, known as the OptoSelect chip right here. And then these antibodies are subjected to screening, and positive hits are then sequenced, cloned, and into an expression vector and expressed recombinantly for testing within various research applications. To help you visualize how this incredible technology works, here are some visual examples. So after cells have been enriched, they are loaded onto a chip as seen here, and within each square is a single B cell being guided into these individual pens known as nanopens. Then each individual well is subjected to one of several assays that help to determine antibody specificity or function. In the video I will show you here, antibodies secreted from a single B cell are able to bind to antigen-conjugated beads located directly outside the well. A fluorescently labeled secondary antibody then helps determine whether antibody binding took place or not. And so here, as you see the beads light up, you can infer that antibody binding has taken place and that the secondary antibody is detecting them. Finally, once the screening assays have been completed and B cells secreting antigen-positive antibodies have been identified, the cells are exported once again with the use of light into collection plates for sequencing and expression, as seen here in this video. And so, what are the types of screening assays that can be done within the chip in order to identify the antibody clones that you want or that are positive for your antigen? In the example I previously showed in the video, the antigen can actually be biotinylated and conjugated to a bead, seen here as an antigen bead. And these beads are then basically secreted antibodies that bind to the antigen directly outside the well can be detected by a fluorescent secondary antibody. More complex assays, such as multiplex IgG capture assays, as well as cell-based assays like the use of GSP reporter cells expressing the target antigen, can also be conducted in-chip in a completely automated fashion. For us, we went with the antigen-specific RBD assay, where we conjugated biotinylated RBD to an antigen bead. In addition to choosing our highly efficient screening and selection method using the Beacon platform, it is also important to bear in mind that the host animal we use for antibody generation is also incredibly important. After all, it is important to start with a host that can generate high-quality antibodies for us to discover in the first place. For us, we chose to use rabbits as our host animal due to their robust and unique immune system that enables the generation of antibodies with superior affinity, specificity, and diversity compared to rodents. GenScript has a lot of experience in generating rabbit monoclonal antibodies using our MonoRab monoclonal antibody generation system. It is a hybridoma-based platform, and with our experience, we saw that we could combine our expertise in rabbit immunization and antibody screening and adapt it for the Beacon platform. In short, what we hope to achieve from this single B-cell antibody discovery campaign was to develop an approach that allows us to really capitalize on the superior performance of rabbit monoclonal antibodies, while also enabling us to achieve more antibody clones with higher diversity in epitope recognition, all in a shorter span of time. And then alongside achieving all of this, we believe that this method will help us address major challenges that researchers may face, not just in SARS-CoV-2 antibody discovery, but in any other antibody discovery project being undertaken. Some of these challenges could be missing out on opportunities due to limitations of hybridoma or display libraries that lead to low diversity or lone clone number in antibodies generated. It could be tied to low success rate if an antigen is particularly challenging and other antibody generation methods have failed. And it also could be tied to challenges associated with just the process of antibody screening and generation being laborious and taking a long time, or being exceptionally prone to errors. I would also like to mention here that GenScript is currently the only vendor that offers rabbit monoclonal antibody discovery using the Beacon system. So how do we go about the process of generating spike RBD rabbit monoclonal antibodies? After rabbit immunization, we generated a single cell suspension from one rabbit spleen, shown here in the second panel, and subsequently isolated plasma cells by subjecting the suspension to antigen and IgG positive B-cell enrichment. Altogether, we imported over 15,000 cells onto the Beacon platform and subjected these cells to antigen positive screening using antigen positive beads conjugated with RBD. Altogether, the isolation and screening and selection process was completed in under 24 hours. And of this, through this process, 142 positive hits were identified, and we exported 60 for sequencing. And so here we go about talking a little bit about the characterization of the antibodies that we discovered. And here we are featuring the top antibodies that we have identified. And so as expected, evaluation of these top antibodies using ViaCore revealed that rabbit mAbs possessed higher affinities than rodent mAbs towards the spike protein. In fact, most rabbit antibodies displayed 10 to 100-fold higher affinity with a KD of 10 to the power of -10 and -11 in comparison to our rodent antibodies that possessed an affinity in about the 10 to the power of -9 range. In addition, epitope binning of several of our top rabbit antibody clones revealed that rabbit antibodies discovered through the Beacon platform were distributed across 4 different epitope bins. And these Beacon discovered clones are in black colored font in this table. In contrast, when we evaluated rabbit antibodies discovered using MonoRab or hybridoma approach, these clones, in red font here, occupied just one epitope bin. Now, while more hybridoma-derived antibodies certainly need to be evaluated for a fair comparison, this observation of higher diversity in epitope recognition is largely in agreement with what the broader scientific community has observed, that a more diverse antibody repertoire can be discovered using the Beacon platform at an efficiency that can simply not be achieved via hybridoma technology. And in addition, the ability to generate diverse monoclonal antibodies that recognize different epitopes on a target are really critical for immunoassay development because this increases the options for identifying antibodies that work specifically in a researcher's application. Furthermore, having diverse antibodies allows for the development of specialized immunoassays, such as Sandwich ELISA, that specifically relies on the availability of antibody pairs that recognize and bind to unique epitopes on an antigen without sterically interfering with each other. With the diverse spike RBD antibodies we discovered using the Beacon platform, we actually identified several really effective antibody pairs, as shown in this table on the left. And we're able to develop a Sandwich ELISA assay that had a sensitivity of 20 picograms per mil towards the RBD antigen, which is a highly sensitive assay at the end of the day. We also further characterize these antibodies for pseudovirus neutralization activity. And so in this pseudovirus neutralization assay, ACE2 overexpressing HeLa cells were incubated with pseudovirus bearing the luciferase gene. And these cells and virus were then incubated also with several different rabbit antibody clones. We then evaluated the ability of these monoclonal antibodies to inhibit viral infection and basically view a reduction in consequent luciferase gene expression within the cells. What we found was that the IC50 values measured from these experiments revealed potent neutralizing activity of these several monoclonal antibodies. Additionally, we went on to test several of our top antibodies for basic research applications, one of which was flow cytometry. We showed just one example here, but actually we do have several rabbit antibodies that work very well in flow cytometry. And so in panel A, the antibody featured here, BSR2B17, was able to successfully sort for RBD expressing cells compared to isotype and PBS controls. Several Beacon-derived rabbit antibody clones were also identified to be well suited for detecting RBD in the supernatants of cells expressing the protein or in its purified form. And so here in lane 1, in the first lane of each antibody tested, we see negative control. In the second lane, we see that these antibodies are able to detect RBD from cell supernatants. And in lane 3, we're able to see that these antibodies are also able to detect purified recombinant RBD protein. At this time, I would also like to mention that all these antibodies I have mentioned so far are available to you for your research. We are very happy to share affinity data, antibody pairing data, as well as characterization information with you. So please do not hesitate to reach out to us. We also have over 70 SARS-CoV-2 antibodies, including nucleocapsid monoclonal antibodies, extracellular domain antibodies, and at any point, our talented account and project managers are able to assist you with any inquiries you have. So please do not hesitate to reach out to us. And so now that I've shown you how the Beacon platform has helped generate some incredibly useful antibodies for your research assay, this begs the question, can we make a great antibody even better? The success of many immunoassays depends on the performance of an antibody in a researcher's specific application. And while an antibody may perform well, is there a way to optimize the antibody for a specific characteristic, such as affinity, to better meet the needs of your assay? At this time, I would like to hand it over to my colleague Win to tell you more about how you can optimize an antibody to achieve its fullest potential in your application. Win, take it away.
Thank you very much, Grace. And hello, everyone. So, Grace, next slide. So now that you have seen my colleague Grace presented how quickly we can generate high-affinity antibodies specific for the spike protein of SARS-CoV-2, I would like to share a case study with you guys to highlight how we can utilize our precision mutant libraries to further improve antibody affinity when needed for specific applications. So in this case study, following the engineering workflow described here, a precision mutant library was designed and built for a lead candidate and subsequently screened for candidates with improved affinities for a diagnostic application. Next slide. So first, what are the precision mutant libraries? Precision mutant libraries are high quality, diverse synthetic libraries containing only desired mutants. More specifically, they contain evenly distributed amino acids with little bias, meaning no over or underrepresentation of specific codons. These libraries also offer inclusion of specific codons defined by users and elimination of stop or unwanted codons, in addition to codon optimization for a specific host of interest for enhanced expression. Our precision mutant libraries are enabled only because of our next generation semiconductor-based DNA synthesis technology, coupled with our automation platform, which together allow us to precisely control incorporation of individual nucleotides into the growing DNA strand on a silicon chip. Next. But in contrast, if you look at the libraries that are commonly constructed through traditional methods using degenerate codons such as NNN or NNK, there are a number of major issues associated with those methods. For instance, there is no control over amino acid ratio. Hence, the libraries are likely going to be biased with over or under representation of certain amino acids. Stop or unwanted codons cannot be eliminated, and codon optimization is impossible in those libraries. So such kind of lower quality libraries requires larger screening efforts to cover the diversity that you want, and thus more cost downstream. With the unique qualities of our precision mutant libraries, as previously mentioned, we can address and overcome all of these issues. Next. So as we are all scientists, here we provide experimental data to support our claim. So we built two libraries and compare them side by side. In this experiment, we also specify a design requirement such that we only want 3 specific codons in the site 1, 10 in the site 2, and 20 in site 3 and 4. As you can see here, on the left side, you cannot incorporate such a design specification in the library built with NNK degenerate codons. You get all 20 amino acids in all 4 mutations, whereas you get exactly what you want specified in our precision mutant libraries on the right side. In addition, if you look at the amino acid distribution, our library is evenly distributed, whereas an NNN or NNK library contains some codons that are under or overrepresented. So this is directly translatable into screening efficiency as shown on the right side, right chart. As you can see here, the efficiency are substantially higher for our library than those of the degenerate library. So as a result, our precision mutant libraries can significantly reduce the burden of screening and subsequent characterization. Next slide. So there are three different types of precision mutant libraries that we offered. The first one is site saturation mutagenesis. This is for those who are interested in mutating a single mutation site to 19 other non-wild type amino acids. The second one is a saturation scanning library, very similar to site saturation, except that now there are multiple sites to be mutated, one site at a time to 19 other non-wild type amino acids. And the third is the Combinatorial mutagenesis library. This is for those who are interested in mutating multiple sites and multiple regions, all at the same time. We build these libraries by synthesizing individual oligos carrying specific mutations and assemble them into double-stranded DNA fragments or clone into a plasmid of choice. We do NGS as part of our QC so that we can provide the amino acid distribution profiles of these libraries. Next. So this is a case study where we targeted 6 different CDRs of an antibody sequence with a total of 63 sites to be mutated to 19 other non-wild type amino acids. This resulted in a saturation scanning library with approximately 1,200 mutants, all in a single pool to be screened. Next. So here is the NGS data for amino acid distribution for each mutation site. Each colored block is an amino acid, and as you can see here, the amino acids are evenly distributed across all 63 mutation sites. We then use our proprietary screening and characterization method to identify top candidates. And in the chart, a value above 1 is considered better than the wild type. As you can see here, there are quite a few candidates with improved affinities generated from our precision mutant library. We then chose a few top candidates for further characterization to quantify their binding affinities. So we use surface plasmon resonance binding assays to determine the KD values, and then found that there were 5 mutations with affinities higher than that of the wild type, with one particular mutation 4 orders of magnitude higher in the femtomolar range, perfectly suitable for a diagnostic as a diagnostic reagent. Next. For the remaining 4 improved candidates, we created a combinatorial mutagenesis library to evaluate the potential effect of combining these beneficial mutations. As you can see here, we found 3 sequences that showed the beneficial combination effects with further improved affinities from those of their individual parents. Next. So currently we're in the works, applying the same strategy using our precision mutant libraries to potentially improve the affinities of a few spike protein antibody lead candidates that were generated from the MonoRep and Beacon platform that my colleague Grace presented. And we look forward to sharing our results in the very near future. Now we'll be happy to answer any questions you might have. And thank you very much for your time and attention.